So we're moving straight into the vaccine session. And sorry, Diana, you'll be the first speaker. Diana is going to talk on smoking and vaccination, and I've messed up your introduction completely, but it's great to have you.
Hi, so my name is Diana Quan, and I'm one of the TB-CRE postdocs working under the supervision of Professor Warwick Britton and Professor Jamie Trickus. And I'm just going to quickly present to you, with the 10 minutes that I have, my current project, which involves examining how environmental impacts, such as environmental factors such as cigarette smoke, impact the efficacy of TB vaccination. So the thing that surprised me, and I'm sure we would all hope for better numbers, is the fact that smoking is still very prevalent today. We are looking at 1.1 billion smokers worldwide as of 2019, according to WHO. And the problem is that 80% of this 1.1 billion live in low and middle income nations. And as we know, that's kind of where the nexus of these high burden country lists for TB, MDR-TB and TB-HIV kind of coincide. So the countries that are boxed in red here report smoking prevalences of over 20% of all adult males. And even the nations where the smoking is not quite so high, we see a lot of use of solid biomass fuel for cooking and for heating indoors. And exposure to this kind of smoke is quite similar to the effects you get from cigarette smoke exposure as well. So we know, and we've known for a long time, that cigarette smoke is quite bad for TB outcomes. We know that smokers have a 9 times increased risk of death from TB, and that there is a strong positive correlation between smoking history and the incidence of latent TB infection. We know that smokers report higher rates of quantiferin false negatives, and in terms of treating a smoker with TB, we see a lot of delayed sputum conversion during treatment, increased length of treatment, increased rate of unfavourable outcomes, and increased loss of patients to follow up as well. But the question is really, how does this occur? Why and how does cigarette smoke exposure impact TB outcomes? And so epidemiologically, we found that cigarette smokers have increased rates of asthma and airway hyperresponsiveness, as well as allergic airway disease and metastatic spread of cancer. But however, conversely, they have reduced hypersensitivity pneumonitis rates. And so together, if we take all of this evidence, it seems as though cigarette smoke exposure is impeding Th1 immunity and promoting Th2 immunity instead. So the question that we're looking at is, does cigarette smoke exposure therefore affect vaccine-induced protection against MTB infection? And if so, how does this occur? So our aims are to investigate this very question, and to do so, we are using a very well-defined mouse model of cigarette smoke exposure. So we are using C57 Black 6 mice, and these mice are exposed to 12 research cigarettes twice a day, 5 times a week, using a nose-only exposure where they're breathing in the smoke mixed with air. And under these conditions, we can get induction of chronic obstructive pulmonary disease quite quickly, within about 8 weeks or so. This model is very similar to what we see in humans. So the mice that are exposed to cigarette smoke exposure using this model show weight loss. They also show increased cellularity in the bronchial alveolar lavage fluid, and also increased cell infiltration in the lungs, and increased production of inflammatory cytokines. We also see thickening of the alveolar diameter, and as you can see in the H&E stains here, we can see that progressive lung damage occurring as the smoke exposure occurs over time. These effects are systemic, so we can see that there is cellular infiltrate in the blood, as well as muscle wastage according to quadricep weight, and enlarged heart size. So that's graphed there, as well as shown in the image. It's quite clear to see when you open them up. And these mice are also more susceptible to infection through infection by S. pneumoniae, as well as influenza. So for our experiment, we have 2 arms. We have a smoking arm and a non-smoking arm. And in each of these arms, we have unvaccinated control mice. we have mice that receive BCG subcutaneously before they start smoking. And then we have 2 doses of our novel subunit vaccine, Sysvac2 Advax, which is administered directly into the lungs as a mucosal vaccine intratracheally. After the 10 weeks of smoking, we challenge the mice with aerosol MTB infection, and then we harvest 6 weeks later to have a look at our protection results. So I'll just quickly present some of our key findings from this initial study with you. One of the things we found is that cigarette smoke exposure does impair mucosal TB vaccine efficacy. So on the left, we have the log CFU per lung, and on the right, we have log CFU per spleen. And as you can see, the mice that were vaccinated with Sysvac2 Advax intratracheally show increased bacterial load in the lungs when they smoke, but not when they do not. We do not see any difference between unvaccinated and BCG groups, except in the spleen, where you can see increased burden in the smoking spleens of the unvaccinated mice. So that's one finding that we are looking to delve into a little bit more. It presents a good opportunity to kind of figure out why this is the case, why mucosal vaccination and not subcutaneous systemic vaccination, why subunit and not BCG. So hopefully by investigating this question a little bit further, we can learn a little bit more about the mechanisms of immunity as well. So some of the things we found that are different. We know that CS exposure alters myeloid cell frequencies in the lungs according to our experiments. So you can see that in the smoking mice, we have significant decrease in alveolar macrophage frequency and increases in interstitial macrophages. So this is kind of at the adaptive level, where the alveolar macrophages are the dominant cell population, which initially engulf and harbor MTB, but they are overtaken by the interstitial macrophages 2 to 3 weeks post-infection, which are quite a stable fraction, which again can harbor and not quite deal with the MTB infection. We also get changes in the DC1 populations and DC2 populations, most notably the DC2s, which are quite potent inducers of CD4 T cell proliferation, which is kind of why we see increased numbers of CD4 T cells, as well as increased CD4 T cell activation. And when we look at the, sorry, I said adaptive before, didn't I? Innate, now it's adaptive immune system. One of the other interesting things we found is the levels of PD-1 expression are quite distinct between smoking and non-smoking groups as well. And PD-1 is a little bit complex when it comes to TB, because if we knock out PD-1 in mice, they die. They succumb to MTB infection quite quickly. At the same time, it's kind of seen as being something that's quite immunosuppressive. So it's kind of like, well, why is that the case? We know that PD-1 is highly expressed on MTB antigen-specific CD4 T cells, and the cells with this expression produce higher amounts of cytokine and proliferate more as well, so it seems like it should be protective. And indeed, the CD4, PD-1 and KLRG1 negative population at the very end is a population that's strongly induced by mucosal BCG vaccination, which is more protective than intradermal vaccination in mice. And you can see that the Sysvac2 group is depressed in that particular population. So that's kind of another angle that we're interested in looking at further. Interestingly, cigarette smoke exposure has little effect on CD4 T cell cytokine production. So the functionality of the CD4 T cells isn't particularly affected despite activation markers, despite PD-1, despite KLRG1. And so that's another thing that kind of confuses this story a little bit and makes us think, okay, there must be something else here to look at. And when we repeat this experiment, there are lots of other things we are interested in looking at. We still have frozen supinatants and fixed lung sections for histology to examine. And in our follow-ups, we probably would also like to look at mucosal BCG immunization, adding another group to compare it to the subcutaneous and see if there's any difference there, if it really is a mucosal specific effect. We'd like to look at more markers of T cell exhaustion, senescence, and differentiation states, since the ones that we looked at before kind of aren't really giving us any clear answers at the moment. It'd be interesting to also look at T regulatory cells and gamma delta T cells, as well as type 1 interferons, because another impact, which I did not show here, is highly increased neutrophils in the smoking mice as well. So that would be another interesting thing to look at. And then finally, in our previous experiment, our initial experiment, we did not look at the BALF or too closely at the MLNs. So that should be another interesting avenue to look at. So I'd like to acknowledge everyone in the TB research program who helped with this research, as well as the Centre of Inflammation, without which the smoking would be completely impossible. And of course, many thanks to the TB-CRE for supporting this work with a seed funding grant. So thank you.
Thank you, Diana, and for catching up on time. But if there's a question for Diana from the audience, maybe just to ask, when BCG was originally given, it was given orally. Is there any suggestion that this is specific to the respiratory mucosa, or is this do you think whether the smoking will have the effect, I would imagine, versus just mucosal vaccine exposure in general? Is there a mouse model of oral vaccine administration for TB?
Orally, I have not seen. I've seen intranasal BCG and intratracheal, I believe. Right. I'm pretty sure I saw a report on that. Not oral, not like pipetting to the back of the throat or anything like that. But yeah, it would be interesting.
Just not for many years. Not for many years, yeah. I know.
A number of years ago that was looked at.
Yeah. All right. Thank you, Diana. That's